through our MCQs. Then apart from that, we have personalized counseling sessions. You can anytime book a slot with us. Then we have three different kind of subscription plans, starting from basic, which is for 3 months, and you have to pay 15K for that. Then followed by standard, which is for 6 months, and then our premium plan, which is for 12 months, only for 50K. You can also get additional 20% off by using my promo code, as you can see on the screen. You will have the benefit of one-to-one personalized guidance and live doubt clarification in premium packages. Okay, so we have certain miscellaneous topics arranged after our protein metabolism, that is TC and ETC. So that was briefly about our schedule. Now let's begin. So taking up a clinical case, a patient presented at day 12 with metabolic acidosis, abnormal urine odor, ketonuria, hepatosplenomegaly. Blood and urine revealed elevated levels of valine, isoleucine, and leucine. Okay. Aggressive treatment was started, including branch chain amino acid restricted diet and supplementation. What is the diagnosis? Okay. So we'll get to know once we're done with our chapter. Okay. So beginning with branch chain amino acids. So we've already discussed that what are our branch chain amino acids? They were leucine, isoleucine, and valine. And the mnemonic for this I had given will. Will is the mnemonic, so you can remember like this: valine, isoleucine, and leucine. Now your leucine is purely ketogenic. Isoleucine is both ketogenic and glucogenic, whereas valine is purely glucogenic. Now all branch chain amino acids, they are non-polar and they are all essential. Okay, if you remember, I told you that the body is capable of synthesizing even the essential amino acids, but it does not, because the step for the synthesis, it requires more than seven or more enzymes, so the body tries to conserve that energy. So but your branch chain amino acids, they are all essential for the same reason, because to synthesize the branches, more than seven enzymes are needed. Okay, to synthesize the branches, more than seven enzymes are needed. So the body prefers to take them through diet. So the body will not spend energy unnecessarily to synthesize these branches. So it is better that you take the branch chain amino acids through diet. So they are supplemented through diet. Supplemented through diet. Okay. Now, the three basic reactions of all branch chain amino acids, they include transamination, oxidative decarboxylation, and FAD-dependent dehydrogenation. Okay, so let's take the individual reactions. Transamination. We've already spoken about transamination. What is happening in transamination? There is transfer of amino group from one pair of amino acid and keto acid to form a new pair of amino acid and keto acid. Okay. So after transamination, your branch chain amino acid, they will become keto acid. Okay. And the coenzyme, we all know that the coenzyme for transamination is PLP, pyridoxal phosphate. Then the second reaction, which is oxidative decarboxylation. So your transamination reactions, they are followed by oxidative decarboxylation reactions. What is happening in this? That the branch chain keto acids, they undergo oxidative decarboxylation. So via transamination, your branch chain amino acids, they were transaminated to their corresponding keto acids. Now these keto acids that are formed in the previous step, they undergo what? They undergo oxidative decarboxylation. Okay, so by releasing one carbon as carbon dioxide. What is oxidative decarboxylation? It is removal. Decarboxylation, it is removal of one carbon as carbon dioxide. Okay, so what does it become? It becomes corresponding branch chain acyl-CoA. And the enzyme involved is branch chain keto acid dehydrogenase. So that is the enzyme that is involved. Then it is followed by fat dependent dehydrogenation reaction. This is your enzyme for this is acyl CoA dehydrogenase. Now, depending upon the amino acid, that whether it is leucine, valine, isoleucine, you get different products. Okay. FAD is which vitamin it is? B2, your flavin. Okay. So let's have a look at the reactions. Let's take the example of leucine. Okay, so you can see leucine. Now, what will happen? It will undergo transamination. So this first reaction is transamination. So transamination reaction, your PLP is the coenzyme. You can see what is happening. This is your first pair of amino acid. So it undergoes transamination to form what? The keto acid. This is the keto acid, okay? And in case of leucine, what is the keto acid? It is alpha keto isocaproate, okay? Now what happens? The next step is your decarboxylation step, which is oxidative decarboxylation. Oxidative decarboxylation. Okay. You can see this carbon dioxide is being removed. So one carbon has been removed as carbon dioxide, this one here. Okay. To form what? To form isovalyryl CoA. Okay. This is your what? Acyl. Acyl CoA is formed. Now the third step is FAD dehydrogenase. FAD dependent dehydrogenase. This is the enzyme. So what happens? You get beta methyl crotonyl CoA. So this is for the case of isoleucine. In case you do not, you don't have to like particularly remember these names. You can just remember the general reactions like you have branch chain amino acid. Okay, now this will undergo what? Transamination. Now on transamination, it will form the respective branch chain keto acids. Okay, now these branch chain keto acids, they undergo what? Oxidative deamination to form their respective acyl CoA. Okay, and this is brought about by which enzyme? Very important. Here, branch chain keto acid dehydrogenase. Okay, so in short, you can write branch chain keto acid dehydrogenase. Okay, and then this is acted upon by FAD dependent dehydrogenase to give different products. Okay, depending on the amino acid. Okay. So in case of leucine, you are getting beta methyl protonyl CoA. Now this will be entering ketogenic pathway. So you see the product formed here is beta methyl protonyl CoA. That's why we say which enters ketogenic pathway. We say that leucine. Here, see, leucine is purely ketogenic. Okay. See, leucine is purely ketogenic. So now you know why. Now moving on to valine, similar reactions will take place for valine as well. This is your valine. It will undergo what? It will undergo transamination reaction to form its corresponding keto acid. In this case, in the case of valine, the corresponding keto acid is alpha keto isovalerate. Then this will undergo oxidative decarboxylation. You can see one carbon dioxide has been removed to form what? Isobutyryl CoA. Okay. Now this will again undergo FAD dependent dehydrogenation reaction. The third reaction to form what? Methacryl CoA. Okay. And this will form what? This will form propanol CoA. Propanol CoA is formed, and we know that this enters the glucogenic pathway. And thus we say that valine is purely glucogenic. See here, valine is purely glucogenic. Now let's have a look at isoleucine. Isoleucine, similarly, it undergoes transamination to form its corresponding keto acid, which is alpha keto beta methyl valerate. Okay, now this undergoes oxidative decarboxylation via the enzyme branch chain keto acid dehydrogenase to form what? Methyl butyryl CoA. Then this will undergo dehydrogenation, which is FAD dependent, to form tigyl CoA. Okay, and then it will split into propionyl CoA and as well as acetyl CoA. Now, propionyl CoA, we know that it has glucogenic path, and our acetyl CoA, it has ketogenic fate. Okay. Therefore, we say that isoleucine is both ketogenic and glucogenic because it is forming in the end both propionyl CoA and acetyl CoA. Okay. So this was about the reactions that the branch chain amino acids undergo. Now moving forward, let's discuss about the branch chain keto acid dehydrogenase. So that is the enzyme which was causing oxidative deamination. That is the second reaction. So this is the enzyme which brings about the second reaction here. This one, this. Okay. So that is branch chain keto acid dehydrogenase. So it is a multi-enzyme complex. It is similar to pyruvate dehydrogenase. Pyruvate dehydrogenase, we know that it is involved in link reaction. So this one is also similar to pyruvate dehydrogenase. Now it has three enzymes, that is branch chain keto acid decarboxylase, abbreviated as enzyme 1. Then it has dihydrolipoyl transacylase E2, and it has also dihydrolipomide dehydrogenase, that is the enzyme E3. So your branch chain keto acid dehydrogenase is a multi-enzyme complex consisting of three different enzymes known as E1, E2, E3. Okay. Then the coenzymes that are involved are CoA, thymine pyrophosphate. Your thymine pyrophosphate is associated with the E1 enzyme, that is branch chain keto acid decarboxylase. Then lipomide, FAD and NAD. These are the coenzymes. The branch chain keto acid decarboxylase E1 is a product of your E1 alpha gene, E2 beta gene. So this enzyme, it is made up of two different gene products, that is a gene known as E1 alpha gene and E2 beta gene. So the product of these two genes, they form your what? They form your beta chain keto acid decarboxylase. Okay. Now your second enzyme E2 is a product of which gene? It is a product of E2 gene. And E3 is a product of E3 gene. So two different genes, that is E1 and E2, they together form these three different enzymes. Once again, I am repeating. Your E1 alpha gene is involved and E2 beta gene is involved. The combination product of these two, they form what? They form your E1. Then E2 is formed by simply E2 gene and E3 is formed by E3 gene. Okay. So the three gene products are forming this multi-enzyme complex, that is branch chain keto acid dehydrogenase. Okay. So two different genes are involved, E1 and E2, and a combination of the products of these two genes, they form your BCKD. Then the E gene defect is associated with the disorder known as maple syrup urine disorder. This is very important. This clinical is very important. You might often get a short note on this. So any defect in any of these genes, it results in a disorder which is known as maple syrup urine disorder. Now let's see what are the different kinds. If the defect is in E1 alpha, So E1 alpha causes a type of MSUD which is known as type 1A. And this is also the most common type of MSUD. Okay. So when your E1 alpha gene is defective, you are having a type of MSUD which is known as type 1A, and this is the most common type of maple syrup urine disorder. Then type 1B, in this your E1 beta is defective. Then type 2, your E2 is defective, and in type 3, your E3 is defective. So you often get MCQs like, let's have a look at this one. MSUD type 1A is due to the mutation of, so now we know that it is due to the mutation of which gene? It is E1 alpha. Okay. Branch chain keto acid decarboxylation is defective in, we know it is defective in maple syrup urine diseases. Okay. Moving forward, maple syrup urine disease. It is usually seen in neonates. Okay. So disease starts in the first week of life. You will start seeing the manifestations within the first week. Now, defect in branch chain keto acid dehydrogenase. So, there is defect in oxidative decarboxylation. So, we know that your maple syrup urine disorder is due to defect in branch chain keto acid dehydrogenase. Now, depending upon the gene that is defective, you have different types, but basically it is, in general, it is the dehydrogenase, branch chain keto acid dehydrogenase, that is defective. So what is being affected? Your oxidative decarboxylation is being affected, and in this branch chain keto acid dehydrogenase also, the most important enzyme, most important, is E1. Okay, so this is the most common, most important defect is in E1 alpha. Okay, so as a result, branch chain keto acid will accumulate and its corresponding branch chain amino acid. Obviously, you see your branch chain keto acids. Okay, they were undergoing what? They were undergoing oxidative decarboxylation to form acyl CoA. Now we are saying that this is not taking place because the enzyme is defective. So this keto acid, branching keto acid, it starts to accumulate, and when this starts to accumulate, naturally its corresponding branch chain amino acid will also accumulate. Now moving on to the clinical features. It is usually present in neonates with feeding difficulties. There's failure to thrive, lethargy, there's acidosis, convulsions, hypotonia with bouts of hypotonia. That is, you have decreased muscle tone. Or sometimes completely hypotonic. Urine on refrigeration for a night gives sweet smell of maple syrup. That is burnt sugar, and hence you get the name of the disease, maple syrup urine disease. Okay, because it smells like burnt sugar. Now, diagnosis, you have increased branch chain amino acids and branch chain keto acids present in urine. Now, different tests for branch chain keto acids, they include dinitrophenylhydrazine test and your Rotharas test. In Rotharas test, we've seen that a purple color ring is formed in the junction. So your Rotharas test is positive. And the other is dinitrophenylhydrazine test. In this, you get a peculiar yellow color precipitate is obtained. Now, treatment includes restriction diet, that is, restrict branch chain amino acids in the diet, and the other is supplement thiamine because decarboxylation of branch chain keto acid, it requires thiamine. Okay, we've discussed this. Seen how here this first enzyme. Yeah. See the coenzymes and the coenzymes. I told you that your E1 is associated with TPP, that is thymine pyrophosphate. So one of the modes of treatment is that you supplement thymine, because decarboxylation of branch chain keto acid requires thymine pyrophosphate. Now, other clinical apart from maple syrup urine is you have another disease, that is isovaleric aciduria. Okay, in this defect is in leucine catabolism. Okay, enzyme defective is isovaleric acid dehydrogenase. It is a FAD dependent enzyme. So you have this enzyme defective. So this is only concerning the leucine metabolism. It has a peculiar smell of sweaty feet. Okay. So severe metabolic acidosis and neurological deficit are seen in case of isovaleric aciduria. There is severe metabolic acidosis and neurological okay. So with this we come to the end of branch chain amino acids. Let's take up a MCQ. Diseases of branch chain amino acids includes phenylketonuria, no. Maple syrup urine, yes. Tay-Sachs, no. Isovaleric acidemia, yes. Niemann-Pick, no. Okay, thanks for watching. With this, we come to the end of our series, that is protein metabolism. If you have any queries, you can ask. Meanwhile, I'll uh, uh, briefly discuss derived amino acids and selenocysteine and pyrrolysine with you guys, since we have some time left with us. So I thought I'll take this up. Okay, in case you have any queries, you can send. So you should know about derived amino acids and pyrrolysine and selenocysteine. You might sometimes get an MCQ on these. So I thought I'll rather not leave it untouched, give you an overview of it. So your derived amino acids, they are derived post translationally. Okay. That is after after translation. Okay. And they do not have a codon. We all know that your amino acids, every amino acid has a codon for itself. Okay. We'll study about translation and codons in the genetic code in details when we take molecular biology. But as for now, for example, you have, let's take the example of AUG. So AUG is the codon for methionine. Similarly, you have like UUU, it might be coding for let's say phenylalanine, some other codon coding for some other amino acid. So you have different codons for each amino acid, but your derived amino acids, they do not have a codon and they are derived post translationally. Okay, now they're classified into two categories, two broader categories, that is on the basis of those which are present in the proteins and those which are not present in the protein. So they're classified into derived amino acids seen in the proteins and derived amino acids that are not seen in the proteins. So which are the ones that are seen in the proteins? They are hydroxyproline, hydroxylysine, gamma carboxyglutamate, your cysteine, methyl lysine, desmosine. Okay. So your hydroxyproline and hydroxylysine, they are present in collagen. Gamma carboxyglutamate, it is seen in gamma carboxylated proteins. Okay. And your cysteine, we know cysteine is, it is formed from two cysteine joined together to form disulfide bond. Like cysteine and you have disulfide bond and cysteine. So this structure which is formed, this is what is known as your cysteine. Okay. So this is also one of the derived amino acids. Then you have methyl lysine. It is present in your methyl lysine is seen in present in myosin. And your desmosine, it is derived from lysine. And where can you find it? It is present in elastin. So these are those derived amino acids that are found in some or the other proteins. Now you have those derived amino acids which are not seen in any of the proteins. They include ornithine, arginosuccinate, and citrulline. I'm very sure that these may be sounding familiar because we've studied them in. They are intermediates of urea cycle. Then you have homoserine and homocysteine. Again, where have we studied them? They were found in metabolism of sulfur containing amino acids. Of sulfur containing amino acids. Okay. Remember your methionine, it was forming SAM, and then SAM was forming homocysteine. So these are broadly the two types of derived amino acids. Now, apart from these derived amino acids, you have two more amino acids that are known as selenocysteine and your pyrrolysine. But these are not derived amino acids. So your selenocysteine, it is not, okay? It is not a derived amino acid. It is the 21st protein forming amino acid and it has a codon. So now you see why it is not a derived amino acids, because I just told you derived amino acids are those which do not have a codon. But selenocysteine very clearly has a codon, which is UGA. So the codon is UGA. Now it is formed by co-translational modification. Another reason why it is not a derived amino acid, because co-translational modification is the is how it is being formed. Whereas your derived amino acids, they are formed after, post-transcriptional translationally. Okay. Whereas co-translational means as and when your translation is taking place, then your derived selenocysteine is being formed. But your derived amino acid, after the translation has taken place, they are then being formed. Okay. The precursor amino acid for selenocysteine is serine. And a process called recoding is involved in its formation. So, how is selenocysteine being formed? It is formed via a process known as recoding. See, what is recoding? Let's suppose this is your mRNA. Okay. This is your mRNA. This is the mRNA head. The 5' UTR. UTR is the untranslated region. Okay. And this is the 3' region. And this is the 3' UTR. That is 3' untranslated region. You must have all studied the structure of mRNA, that mRNA at its ends at the 5' end and the 3' end has the untranslated regions, and in the center it has the coding part. Coding part is the part which codes, okay, and the extreme ends, they are not involved in coding. So what happens is, when the mRNA at its 3' UTR, it has a structure like this. As you can see here, this structure, a structure like this, known as stem loop structure, known as sequence. Now, so if the mRNA at the 3' UTR has a stem loop structure which is known as sequence. What is sequence? It stands for selenocysteine insertion sequence. Okay. So if sequence is present at the 3' UTR, then what happens? That it changes the UGA, that is the stop codon in the coding part to coding codon. And this process is known as recoding. So what is recoding? Recoding is that your UGA, that is the stop codon, it changes into coding codon. This process is known as recoding. So what is happening is your sequence, what did it do? It converts stop codon to coding codon. And this is known as recoding. So now what happens? Now your UGA, what will happen? A tRNA which is carrying serine will come. Okay, now serine, serine has hydroxyl group. Now this serine was first initially changed into cysteine, which will be having sulfhydryl group. Now sulfhydryl group you have to ultimately change into, it should have a selenium. Okay, so now selenocysteine is formed when the sulfhydryl group is changed into selenium. So this is how stepwise your selenocysteine is formed from serine. That is why we say the precursor amino acid is serine. Even if you're not able to get certain part of this, it's okay. But basically, you should have a little bit brief idea about selenocysteine, that it is not a derived amino acid. You might get these in MCQs. Okay, you should know, at least if you're not able to understand the entire recoding step, you should know that what is the process by which selenocysteine is formed. So it is formed via recoding. Now, although I've tried to explain you, you have a little brief idea that what recoding actually is. Recoding is simply, if I tell you that when sickest element is present in the 3' UTR of an mRNA, then it changes, it converts the stop codon UGA into a coding codon. Okay, so this is simply what recoding is. So your selenocysteine is formed via a process which is known as recoding, and the precursor amino acid is serine. So serine then changes into cysteine, and then cysteine changes into selenocysteine. And this is a co-translational modification. You see, all of this is happening while translation is taking place and not after. So this is not some post-translational modification. Okay. Now some examples of selenocysteine. So selenocysteines basically they are present in the reductases and peroxidases. Okay, like your glutathione peroxidase, glycine reductase are some examples for it. These enzymes they contain selenium in its active site. Okay, they contain selenium in its active site. Okay, talking briefly about pyrrolysine, it is again not a derived amino acid. It is very similar to selenocysteine. It is the 22nd protein forming amino acid. The codon for pyrrolysine is UAG, and it is also derived co-translationally. The precursor here is lysine. Okay, so the precursor amino acid for pyrrolysine is lysine. It also comes from the name itself. Now, this is seen in bacteria. So, your pyrrolysine is not seen in humans. You might get an MCQ that which of the following amino acids are not seen in humans, and you might get 4 different options: pyrrolysine, selenocysteine, hydroxyproline. Okay, so your answer will be pyrrolysine, and the codon for this is UAG. So, with this, I think we've now covered almost everything that comes under amino acids and its metabolism. So this was a small topic which I thought you should know. So with this, we come to the end of our series on protein metabolism. Hopefully, we'll be starting with tricarboxylic acid cycle, that is TCA cycle, Krebs cycle on Monday, followed by electron transport chain. Very important, both of them. If you have any queries, you can send them in. Okay, thanks for watching.